So we've seen that osmosis is important in determining the distribution of water within the body's fluid compartments. So recall that these fluid compartments are compartments within the cells, the blood, the interstitial fluid, and things like that. So clinically, you're going to encounter patients with swelling or fluid swelling, which is also known as edema. And edema is caused by abnormal water accumulation and retention within one or more of these fluid compartments. We've also seen that if a solute concentration on the two sides of a plasma membrane are different, the water concentrations are going to differ too, because solutes take up space that would otherwise be occupied by water. So when solutes are dissolved in water, they don't just disappear, they instead take up space or volume that would otherwise be occupied by water. So the more solutes we have on one side of the membrane, generally the less water we're going to have because there's less space for that water to take up. So we saw that this is a pretty defining characteristic, I should say, of diffusion, or what drives diffusion, I should say. So water, we saw, will also tend to move by osmosis towards the side of the membrane or towards the fluid compartment with the lower concentration of water. And so recall that osmosis is a form of facilitated diffusion, and like all forms of diffusion, a substance, in this case water, is going to move from an area where it is more highly concentrated to an area where it is lower in concentration. So we saw that osmosis, being a form of diffusion, works just like the other forms of diffusion as well. And because water moves and water molecules also take up space, that means that if water moves from one compartment to the other, the volume, or the total volume of that compartment, will change as well. So here we're actually going to look at the consequences of the movement of water and the volume changes that result as water moves from one compartment to another. So let's illustrate this by looking at a simple example of how water will move by osmosis and how that movement of water will change the volume of the compartments within the body. So our simple example here, we're going to take two vessels, and both of these vessels have the same total volume, which is 1 gallon here. We'll work with 1 gallon for now, because that's a volume that we're all familiar with. In the left vessel here, we have 1 gallon of pure water. So there's only water in this vessel. So that 1 gallon of fluid is taken, or that 1 gallon of volume is taken up by 100% water. In the right vessel here, we've placed table salt into that vessel, and that table salt amounts to 10% of that volume of that vessel on the right here. So we've filled this vessel with 10% of its volume as table salt, and then we'll add water to it until we reach 1 gallon. And so what that means is that when this table salt dissolves in the water here, this table salt will take up space that would otherwise be occupied by the water, so it just doesn't disappear. So as a consequence, there's less water in this vessel on the right here, because 10% of the volume of this vessel is occupied and taken up by table salt, which means that 100 minus 10, the remaining volume that we have that's water, is 90% water. So what we see here is that the concentration of water is much less in this vessel on the right, and there is a higher concentration of water in the vessel on the left here. So we've got our two vessels here now, and we've kind of established what's in all of them. Now let's kind of simulate the fluid compartments in the body, or at least the movement of water between fluid compartments in the body, by taking a simple membrane and placing that membrane between these two vessels here. So what we're going to do is we're going to put a water-permeable membrane in between these two vessels here, which this water-permeable membrane is represented by this orange bar here. So my questions for you at this point are going to be, which direction will water diffuse and which direction will salt diffuse? Now, I have to apologize because this is a little bit of a trick question here. You have to pay attention to or you want to note the limits of our membrane here. So this is a water permeable membrane, and that means that only water can move across this membrane. So that means that water will diffuse across the membrane, but salt is going to stay put. That is, this membrane is impermeable to salt. So as a result of that, water is going to diffuse from the left vessel to the right vessel, because recall that there is a higher concentration of water on the left side than there is on the right side. And so we're going to get diffusion, aka osmosis, of water from left to right here. But remember that our membrane is selectively permeable. It's a water permeable membrane, but not a salt permeable membrane. So as a result of this, salt does not diffuse. Salt stays put. Right? So we saw that water moved, but salt did not. Right, in this particular example. So what's going to happen to the volume in each of these containers here? Well, as you probably guessed, because water moved from the left vessel to the right vessel here, the volume of water in the left vessel will decrease, and the volume overall in the left vessel will decrease, and the volume overall in the right vessel will increase. Right, so we're going to get a volume change across this membrane, and that's pretty analogous to the way that cells behave, right? So our simple example there that we used with those two containers is a pretty accurate model of how cells in our body behave, because what cells like to do is they like to hang on to a certain number of solutes, or a certain concentration of solutes like salts, sugars and things like that, proteins within their cytosol. And those things are generally not as easily able to cross the plasma membrane as water is. And so cells like to hold on to their solutes. And what we get is we get water moving into and out of the cell as a result of changes in concentration of those solutes. Right. So we saw that in our simple example with our 1 gallon vessels there, we saw that the volume on both sides of that membrane changed when the water moved. And as I mentioned, this is pretty analogous to the way that cells behave. This is actually just another example. This one's straight out of the textbook, but it is just a different way of looking at the two vessels situation or simulation that I had set up in the previous slides here. So we've got this U-shaped channel here. We've got two equal volumes of solution and solute, or I should say equal volumes of material on both sides of this U-shaped channel, and we have a selectively permeable membrane between the two. And these red dots within it represent a sugar in this example. So these are solute molecules which are represented by sugar. And this selectively permeable membrane is only permeable to water but not sugar, which means that water is going to diffuse from this side of the left side of this U-shaped tube here to the right side, because there's a higher concentration of water over here and a lower concentration of water on this side. And so we're going to get volume changes. The volume on the left side of the channel or U-shaped tube is going to decrease. The volume on the right side of the U-shaped tube is going to increase. Right? So we got a volume change as water moved. So here I have a few questions for you to answer, and I will post the answers to these questions in the announcements forum on Moodle so that you may check your work. The simulation or scenario that I have, maybe scenario is a better word for you here. This scenario that I have here for you is that you have two solutions, A and B. So solutions A and B are separated by a selectively permeable membrane. Right? And we see that you make an observation that over time, so you let this semi-permeable membrane sit, you have kind of no idea really what's in solutions A and B. You just know that there are two solutions. And you make an observation that over time, the fluid level inside of solution A increases. So my questions for you are this. So which solution, A or B, initially had the higher concentration of solute? Which solution, A or B, had the higher concentration of water in it, and give a simple explanation for both of your answers. As I mentioned, I'm going to post the answers to these questions in the announcements forum on Moodle so that you may check your work. And if you have any questions about your answers and the answers that I give you, please feel free to email me or contact me, and I'd be happy to help you work through these problems here. Okay. So now that we have thought about water movement and changes in volume, let me introduce you to the concept of tonicity, right? So we have just learned that changing the concentration of solutes or water on one side of a membrane is going to promote osmosis of water across that membrane. And that osmosis, or that movement of water, is going to result in volume changes here. So let's look at this concept of tonicity. Now, tonicity refers to the ability of a solution to change the shape of a cell by altering the volume inside of that cell. And with tonicity, we see that there are three possibilities. The solution, our solution in question here, could cause the cell to lose water. Our solution in question here could cause the cell to neither gain nor lose water, or our solution in question here could cause the cell to gain water. So let's look at all three of these scenarios one by one. So first I'll introduce you to the isotonic solution. So if we were to place a cell, so in this example we have a red blood cell into an isotonic solution, which is this is our simple little our beaker set up here, and we've placed our cell into our solution here. If we place this cell into an isotonic solution, we would find that the rate of water gain by the cell is going to equal the rate of water loss by the cell. So as you can imagine, this cell is going to retain its normal shape and volume because water gain by the cell is equal to water loss. So remember that diffusion, I want you to keep in mind that diffusion is driven by the random movement of particles. So in this isotonic solution, there will be a random amount of water gained by the cell, and that random water gain is going to be balanced by a random loss of water from that cell. So this, over time, this cell will neither gain nor lose, or I should say, in general, this cell will neither gain nor lose water. So it will gain and lose water at equal rates, right? So gain will not be greater than loss, or loss will not be greater than gain. Hopefully that's clear enough here. So in this example diagram here, we've placed our cell, our red blood cell, I should say, into our red blood cell contains about 0.9% saline within it and about 5% glucose. So what I want you to do here is, because we're thinking about osmosis, right? Osmosis has to deal with the movement of water. So what I want you to do when you encounter a situation like this and you're thinking about osmosis is that I want you to calculate the amount of water that's in the cell, and I want you to calculate the amount of water that's in the solution that you've placed it in. So here I've given you that the cell is 0.9% or 0.9% saline and 5% glucose. I want you to assume that the remainder of the volume in this cell is occupied by water. So let's now place that cell into a beaker that also contains 0.9% saline and 5% glucose, because this is our isotonic solution. This prefix iso means same, and we can see that because the concentration of solutes inside of our cell is equal to the concentration of solutes in the solution that we placed it in. So iso, same, so it's the same tonicity. So we've placed our cell into a beaker that contains 0.9% saline and 5% glucose. And so assume again that the remainder of material in that beaker is water, right? Because remember that tonicity deals with the solution's ability to move water. So what we're going to want to do is calculate the amount of water in each of the fluid compartments, and then see where those water concentrations are the lowest, because osmosis is always going to move water towards the fluid compartment where water concentrations are lowest. So what we see in this example here is that if we subtract 5 and 0.9 from 100%, right, because the volume of something is 100%, so that is, you always start with 100% as your total volume, and then you subtract the components of it, right? So if 5% of that total volume is taken up by glucose, you subtract that from 100 because that's the the total volume of our, that 100% represents the total volume of our cell or volume of solution in this case. So subtract 5 from 100 and 0.9 from 100, and those give us 94.1% water inside of the cell. And because we placed it in isotonic solution, there is 94.1% water outside of the cell. And so in this example here, we see that there is no location in which water is lower because water concentrations are equal on both sides of the membrane. So water is going to move randomly either into or out of our cell. And so therefore the gain of water will equal the loss of water in this example. Okay. So we tend to see clinically that drugs that are given intravenously right into the veins are usually placed or dissolved into a solution that's called lactated Ringer's solution. And lactated Ringer's solution is a type of solution that's specifically calibrated with solutes and sugars and things like that, because lactated Ringer's solution is an isotonic solution to the blood that you're placing it in. So we want to be able to deliver that drug without causing any osmotic stress in the body. And so we place intravenous drugs into lactated Ringer's solution, and then we put that into an IV and administer that drug and solution intravenously. Right. So let's look at, since we've looked at isotonic solutions, now let's turn our attention to scenarios in which the volume of the cell will change. And the first one that we'll focus on here is the hypertonic solution. And this is where the cell is going to lose water to its environment, because in hypertonic solutions there are more solutes outside of the cell than there are inside of the cell, right? So in our example here, we placed our cell into a solution that contains 5% saline and 10% glucose. So remember to subtract 5 and 10 from 100, right? Because 100% of our volume minus 10% of that volume is glucose, 5% of that volume is saline. So that means that the remaining volume of our solution here is water, and the remaining volume is 85% because we had 100 minus 10 minus 5. And so that means that there is 85% water outside of our cell. And inside of our cell, we still have the same concentrations that we saw before, which is 5% glucose and 0.9% saline. So this means that our cell still contains 94.1% water. So there is a lower concentration of water outside the cell than there is inside. So this means that water is going to diffuse by osmosis out of our cell, and our cell will tend to lose more water than it gains. And what this does is it causes the cell volume to shrink or crenate. And so this is an osmotic stress on the cell, and it's going to cause water loss, and the cell will shrink or crenate. Now, on the other hand, we have the hypotonic solution. So be careful to not confuse these hypertonic and hypotonic, because they both sound very similar. They're both spelled fairly similarly, but they have the complete opposite effect on the cell. Now, in hypotonic solutions, there are less or fewer solutes outside of the cell than there are inside of the cell. And so once again, in our example here, we have placed our cell into a solution that contains no saline and no glucose. So we've basically placed it in pure water, which means 100% of the volume outside of the cell is water. And because our cell has 5% glucose and 0.9% saline inside of it, this means that there is 94.1% water inside of our cell. So this means that our cell is going to gain water from the solution. That is, there's a higher concentration of water outside, lower concentration of water inside. Water is going to diffuse into the cell more than it diffuses out, right? Because remember, diffusion is a random process. So we will have some water loss in hypertonic, or I should say we will in hypertonic solutions, we'll have a little bit of water gain, and in hypotonic solutions, we'll have a little bit of water loss, right? But overall, in hypotonic solutions, we're going to have more water gain than water lost here. And because our cell gains water, that's going to cause the cell to swell up. And if the cell swells enough, it could actually burst or lyse. And so the hypotonic solution is still causing an osmotic stress on the cell, but it has the opposite effect that a hypertonic solution has. Hypertonic causes a decrease in cell volume, the cell shrinks. Hypotonic causes the opposite type of osmotic stress, and we're going to see that our cell is going to swell and could potentially burst, which of course is not good for the cell, because if the cell bursts, that means the plasma membrane is ruptured, and then there is no longer a separation of environments, fluid environments, between the cell and its external environment, or its the fluid or solution that it's in. So if the cell bursts, that compromises the integrity of our plasma membrane.